Hello, this is Mrs. Kohler, and this is my buddy Tenchi. He's going to help me today. And today we're going to talk about meiosis. We've talked about mitosis, but see, I have an egg here. Meiosis is the process that creates eggs and creates sperm. See, normally when a cell divides, you get the normal amount of DNA in it that's just identical to the parent. But this would be a problem. If you have a sperm and an egg combined and they both have the total number of DNA, then you have twice as much as you need. So what we need is a process that produces half as much DNA so that when the two combine, you have the regular amount of DNA. So that's what we're going to be doing today. So we're going to be finding out how this egg is made. Say bye, Tenchi. Now today we're going to talk about the stages of meiosis. What happens at each stage? So first, why do cells do meiosis in the first place? Well, they do it to cut the number of chromosomes in half, and that's to produce gametes, or sex cells, for reproduction. So here's a diagram of meiosis, and you can see all of the different stages. There's a lot more than in mitosis, but what you can see is that the cell divides twice. The first time it divides here to make 2 daughter cells, and the second time each of those divide to make 4 daughter cells. So that's going to be one of the big differences between mitosis and meiosis. Now, because the cell divides twice, we call these stages meiosis 1 and meiosis 2. So let's talk about meiosis 1. In meiosis 1, genetic recombination occurs. Remember, the two types of genetic recombination were crossing over and independent assortment. These are happening in the first half of meiosis. The homologous chromosomes are going to separate. Those one from mom and one from dad, they're going to pull apart and go into different cells during meiosis 1. So, just like in mitosis, it starts with interphase. So it goes through G1S and G2 just like normal. So your sketch for interphase is going to look just like your sketch for interphase for mitosis. Now, in prophase 1, because this is preparing for the first division, the chromosomes condense just like in mitosis. But now you have an extra step. You have the tetrads forming and you have crossing over occurring. So when you do your sketch, make sure that you show you have your 2 sets of homologous chromosomes and show that they're crossing their arms over each other when you do your sketch. Because the tetrads forming and the crossing over is very important. Now, metaphase 1 is close to metaphase of mitosis, except that instead of individual chromosomes, instead of a single line down the middle, now you're getting a double line because you have that whole tetrad moving. And the chromosomes are going to assort independently, which means some of moms are going to go in one direction and some of dads are going to go the other direction, and you never know who's going to go where. So here, instead of a single line of chromosomes, you have a double line of chromosomes. So that's metaphase 1. And see how here mom's chromosomes, one chromosome is on the left and the other chromosome is on the right? That's independent assortment. In anaphase 1, remember A for apart, you have chromosomes pulling apart, but the centromeres aren't coming apart like they did during mitosis. You have the actual tetrads breaking up. So the homologous chromosomes are separating. And the sister chromatids are going to stay together. So here you have entire X's going toward the sides instead of individual chromatids. And then telophase 1 and cytokinesis, you get your formation of 2 nuclei, and cytokinesis occurs, so the cell divides in half, and I'm just lumping them together even though that you know they're 2 different stages. So here you have 2 new nuclei, each one with a full chromosome, but if you'll notice, because those blue and green chromosomes exchange some material, each chromosome is different now. Now, quick review before we go on to meiosis 2. How are prophase 1 and prophase of mitosis different? Well, in prophase 1, tetrads form and crossing over occurs. They don't happen during mitosis. Why do crossing over and independent assortment occur? Well, that's for genetic recombination, so that the offspring have genetic differences. Make everybody unique. interkinesis. Inter kind of means in between, and kinesis is sort of like cytokinesis. This is sort of like an interphase in the middle of meiosis, except that there's no DNA dividing. So it's more like a resting phase. So the chromosomes relax, they don't completely unwind, but you have a brief resting phase for the cell to get reorganized. And also, because half of your centrioles went each direction, you need to replicate those so that you have 2 sets for the next stage. Now, meiosis 2, which is the second division, what happens? Well, the sister chromatids separate. It's now occurring in 2 cells instead of 1, but other than that, it's pretty much exactly the same as mitosis. So prophase 2, the chromosomes form again. Now this is occurring in 2 cells instead of 1. So basically what you have is 2 cells that look like they're in prophase of mitosis. Metaphase 2, chromosomes line up in the center, and it's just 2 cells instead of 1. So now you have your single line, you just have a single line in 2 different cells. Anaphase 2, sister chromatids come apart just like they do in mitosis. So now you have individual chromatids instead of the full X's, but it's happening in 2 cells. Telophase 2 and cytokinesis, you now have 4 new daughter cells, and each of these cells is haploid. Remember, the cell started out diploid with 2 sets of chromosomes. Now it's ending up haploid with 1 set of chromosomes, and each cell is genetically unique. So when you look at these chromosomes, no 2 nuclei have exactly the same genetic makeup. They're all a little bit different. Now, another quick review. Key differences between meiosis 1 and meiosis 2. Well, in meiosis 1, you get genetic recombination and the homologous chromosomes separate. Meiosis 2, no genetic recombination and the sister chromatids separate. Now, stages of meiosis 2 compared with mitosis, well, they're almost identical, except that meiosis 2 is occurring in 2 cells instead of 1. Number of chromosomes at the end of meiosis, with the number in the parent cell, half the original number of chromosomes, it's haploid. How do prophase 1 and 2 differ? Well, in prophase 1, you have crossing over, and it's occurring in 1 cell. Prophase 2, no crossing over, and it's happening in 2 cells. Anaphase 1 and 2, how are they different? Well, in anaphase 1, homologous chromosomes separate. Anaphase 2, sister chromatids separate. Now, I'm taking the trouble to go through these on the PowerPoint. You'd think they might be test questions.